Hey guys, so here is our organic and biochem homework. It's the last homework in 112, so I'm sure you're happy about that. So I'm just going to start out with question number 1. All right, so a little reminder, when you have, when you're practicing this stuff, I really recommend printing out your pamphlet that you get on your final exam. So again, this handout has a nice guide for what an alkane is. So it has an example of all single bonds, right? A name and the generic formula. So this allows you to say, tell me how many hydrogens would be attached to a 6-carbon alkane or a 6-carbon alkene or an alkyne. So like that. That's a handy thing to use. For this worksheet, this organic functional group chart is a helpful thing, table is a helpful thing to use. Okay, so in this case we have this structure, and it's not showing us the entire structure. Oops. It's just showing us the functional group in question, the C double bond O. We call this a carbonyl group, and then it says that carbon has 2 hydrogen attached to it. So I'm looking for something that has a carbonyl with hydrogen. So let's see, halides don't have any carbonyl, alcohols don't, ethers don't. Here's a carbonyl, and it has an H attached to it. And now, as we discussed in class, this R could be anything. It could be another carbon or a hydrogen or anything like that. This is an aldehyde, so that's different from a ketone where you don't have a hydrogen attached to this carbon. You have other carbons attached to it. Carbonyls also occur here in the organic acids, which we also call carboxylic acids. So you have the carbonyl with an OH, that's carboxylic acid. An ester is just the carbonyl with an O on it. And then finally we also have carbonyls attached to amides. So basically if you take an amine, which is N with anything else on it, so NH here, and you attach it next to a carbonyl, that's what an amide is. So carbonyls happen a lot in organic chemistry. They're a very common thing. Ooh. Okay, so based on our little chart, we just figured out that something where you have a carbonyl and 2 hydrogens attached is going to be an aldehyde. Okay, so as for the next one, here we have some hydrocarbon, carbon, hydrogen, and then another carbonyl group, and again with the H on it. So it doesn't matter what all this stuff is, whether it's just a hydrogen or a whole bunch of other things. Like in this case, it would, have essentially a butyl group attached to it. So it's still an aldehyde because it has that carbonyl with an H. Over here we have a carbonyl again, but this time there's no H attached to it. So when we look on our chart, that looks like this, a ketone, right? So it's just a carbonyl in the middle with carbons on both sides. So that's a, oops, sorry, I hit the wrong button. That's a ketone. And down here, let's see, the red, it might not be red on the printout I gave you guys because it was black and white, but on the digital document, the carbonyl is shown in red because oxygens are generally represented as red. So this is the functional group again. This time we have 2 carbons on either side, so this one is also a ketone. Alright, so now the next kind of question is kind of go the other way. We're starting with a name, we got to draw a structure. So propanone, okay, so prop always means the same thing, and if you can't remember what it means, use your handout, right? So the prefixes are listed on the page prior to our organic functional group. So prop always means 3. So we're going to start out with 3 carbons in the chain. And then, so propanone, O-N-E, I'm going to go back to our chart here. O-N-E refers to O-N-E pentanone, right? So O-N-E is the ending for a ketone. So I'm going to put a carbonyl in the middle of this compound because that's what a ketone has. And then I got to fill in my hydrogens. You could draw them as like sticks with an H on it, or you can draw it as CH3. Butanal. Okay, so let's see. Al is the ending there. So we look on our chart, and oh, I went too high up. Al happens in the example name propanal, right? So that means it's an aldehyde. Al, aldehyde. It's interesting. Propanone has ketone in it. Butanal has aldehyde, right? So the ending kind of sounds a little bit like the functional group. Oh, these all say A. That's not right. Okay, so obviously this is B. So butanal, but means 4 atoms, so we're going to do 4 first. An aldehyde always happens, the carbonyl happens at the end or the beginning, either one is fine, because it has to have an H off of the carbon. And if you put a carbonyl in the middle, sort of like this, there's not enough bonds to put a hydrogen on that carbon. When you have aldehydes, the first carbon is always the one with the carbonyl on it. It doesn't matter whether I draw it at the end or the beginning, right? Because, again, we can number backwards, and that's okay. All right, so now we've got all of our H's drawn in perfectly. This one, hexanone. I always kind of start at the end of the word, because, of course, that's the parent chain. All right, so we start at the end here. So hexa means 6, like hexagon. None means ketone, right? So this carbonyl is going to be somewhere in the middle of the molecule. Now we have so many carbons to choose from that we do need to number where that ketone occurs. So that's what this 3 means here. So we're going to put the carbonyl on the 3rd carbon, right? Up here, we don't have to say 2-propanone because that is the only place to put a ketone in. You couldn't put it on the end, then it would be an aldehyde. All right, so, and then it also says 2-methyl. We know that a methyl is a single carbon group, so we're going to put that on the second one. And I'm just going to fill in the rest of my hydrogens. Remembering, of course, that each hydrogen needs 4 bonds. Okay, so that's 2-methyl-3-hexanone. Lastly, on this question, we have pentanone, so that's 5, penta, like pentagon. Okay, so 2 refers to where we're going to put the carbonyl. It's pentanone, so it's in the middle, so 2. And 3-ethyl. Now, an ethyl group is 2 carbons hanging off, so I'm going to write like that. That's 3 bonds, so I'm going to add a hydrogen in there, and that's only 1 for the end one, so add 3 more. All right, so for the next question, so when we're thinking about boiling point, that always goes back to the chapter where we learned about intermolecular forces. So intermolecular forces, so just a really fast review of that, and if this is too fast for you, I really recommend you go back to the chapter where we learned about these. So let's just do it in order, I suppose. The weakest intermolecular force, which is the one that everything has, every single chemical has this force, is called London dispersion forces. London dispersion forces are weak, meaning easy to break. Compounds that have only London dispersion forces will have very low boiling points, low melting points. They'll be very volatile, right? So have high vapor pressure. All of the properties we learned about in that chapter come back into play when we talk about organic molecules. Organic molecules tend to have a lot of London dispersion forces. Carbon-hydrogen kind of connections are the most common London dispersion chemical, or part of a chemical, really. We learned when we talked about London dispersion forces that size matters. The bigger the molecule, the more London dispersion forces that it will have. By the way, sometimes we call this LDF, right? So if we have a really long chain of carbons, such as you would have in a fat, let's say, it's going to have really high London dispersion forces. And this kind of competes with all the other types of IMF because all of the other types are charged particles, right? So there's kind of like a division here. If you have something with LDF as its dominant, meaning most important, intermolecular force, then it's unlikely it's going to want to bond with something that has a dipole, for example. So the next strongest IMF is dipole-dipole. Then we have a special kind of dipole-dipole that is so common and so strong that we made, oops, I don't need to keep writing the word bond again. We made another category out of it, right? So there's hydrogen bonds are a kind of dipole where you have an H connected to a really electronegative atom. So the three most electronegative atoms in the periodic table are fluorine, nitrogen, and oxygen. Some people remember this by remembering if you have hydrogen that is Fon, get it? It's hilarious. Then that would be a hydrogen bond. Finally, our strongest intermolecular force is when you have ion-dipole bonding. So this is when you've got an ionic compound that's dissolved in something with a dipole, such as like salt into water. Okay, all three of these are charged here. That's why they're so strong, positives and negatives. Dipoles are weaker because they're partially charged. H-bonds are partially charged, but more separated, more polar. And ion-dipole are completely positive or negative, so that makes that the strongest interaction. LDF is left here all by itself. Things that have predominantly LDF forces only like to mix with other things that have LDF forces. That becomes really important when we're talking about organic chemistry. Okay, so back to the actual question. Here we have a comparison between three pentanone. So none means ketone, right? So a ketone, I don't know, I guess it could help to just draw the structure here. So pentanone has 5 carbons. The third one having our ketone on it, right? So I'm not going to draw the hydrogens in here, but that's, you know where they are. And versus 2 pentanol. So again, penta means 5 carbons, so that part's the same. 2 refers to where this all occurs, and all, OL, is from alcohols. So alcohols, again, it's on our sheet if you forget, but OH is the key there. So we're comparing 3 pentanone to 2 pentanol. And so to make these kind of comparisons, we need to decide what kind of IMF. All right, so the ketones are all going to have this carbonyl bond, and oxygen really likes electrons, so that's partially negative, and carbon doesn't really like electrons, so that's partially positive. So here we have a good dipole-dipole situation. Over here, there's still a separation in charge here, but the more dominant thing, the bigger energy here, is going to be from hydrogen bonds. Okay, so what we have here for the next one is ethanal. So Al makes me think of aldehyde. And ethane, right? And again, if you don't remember the endings, these words, use the handout that you get on your final, okay? So eth means 2. Al means aldehyde, so it's going to be, well, ethanal can only be an aldehyde if you're going to, between ketones and aldehydes, it can only be an aldehyde because there's nothing in the middle, right? It's going to be a CH3. So this is ethanal. Aldehydes have that wonderful carbonyl group again, so it's going to be partially negative, partially positive. So this is a polar group. They're asymmetric, so it's a polar molecule as well. So that means that it's going to be a dipole-dipole. Oh, my eye got lost in there somewhere. Okay. Ethane just means 2 carbons with all the hydrogens that it can fit, right? Because alkanes are all single bonds. So this one has no oxygens in it, it has no nitrogens in it, it has nothing to do hydrogen bonding with, and it doesn't have any kind of asymmetry. It's perfectly symmetric. That's a tetrahedral carbon and that's a tetrahedral carbon, so they match. So since it's symmetric, this molecule is nonpolar, which means that its force is LDF. We are looking for the stronger force, right? Because stronger force means higher boiling point. So the stronger force is the dipole. So we're going to choose ethanal. Okay. Propanol, all OL means alcohol. So we're going to go 3 carbons, and then we're going to plop an alcohol on it. It says 1 propanol, so this would be your first carbon here, right? And then all the hydrogen fill in. So this would have some LDF because, of course, you've got hydrocarbon hanging out. But more importantly, stronger than that, is the OH. And that, of course, is hydrogen bonds. So all alcohols can hydrogen bond. Propanone has 3 carbons and a ketone in the middle. This is a dipole-dipole again. Ketones always have a dipole-dipole. Aldehydes also, same thing. Alcohols always have OH, right? So these are kind of trends you gotta learn to recognize. Propane is 3 carbons. Nothing else fancy about it. Pentanal is going to be 5 carbons. So it's got more LDF in it because it has more carbons in the chain. And then the Al means it's an aldehyde, so we're going to put that carbonyl on the end. So pentanal has more LDF, but more importantly, even stronger than that, is that this is a dipole-dipole as well, so that's going to have a higher boiling point. Which, I don't know, I probably could have anticipated that just because propane is a gas at room temperature, right? You use that for your gas grill. Oh, this is supposed to say versus. There we go. So now we're looking for higher aqueous solubility. So the trick to solubility is we are looking for something with the lowest amount of LDF, right? Because non-polar things don't mix that well with water. Or maybe and is better. Highest, like IMF in general. Okay, because of course LDF is the lowest thing. So, all right, so we just draw these. So heptanone has 7. 1, 2, 3, 4, 5, 6, 7. It says 2 heptanone, so that means the ketone goes on the second carbon. And propanone we've seen several times now. It's just 3. So they both have the dipole-dipole interaction, so that part is the same. That means that they will interact with water to some extent. The longer the carbon chain, the less soluble, because we have more LDF here. So this one has more LDF than the other one does. So just 3 carbons versus 7. So the fact that it has more LDF means that it's going to be less soluble in water. So we're going to pick the propanone being more soluble in water. Okay, so pentane here is going to be 5 carbons. Nothing else, just 5 carbons. Pentanol is still 5 carbons. So at least the LDF will be the same for this one, for that carbon chain. And it says pentanol, so that's going to have an OH. Now the OH could go anywhere on the molecule because it doesn't tell us what number, so this is not a proper name. But it doesn't matter where you put it. The bottom line is you have the same amount of LDF for both of these, right? Relatively high, lots of carbons. But this OH on there is going to make the pentanol dissolve in water better. So we're going to pick that one. And pentanone, so again 5 carbons. 3 pentanone means I'm going to stick that carbon in the middle. And 2 pentanol, so again, pent, same, right? So if it's 5 carbons, they're going to have the same amount of LDF. This has an OH on it. Okay, so this is dipole-dipole, and this is hydrogen bonding. Hydrogen bonding dissolves in water better. Again, higher IMF. Alright, so here we are identifying biochemical molecules. And we learned about actually 5 different categories of biomolecules in class this week. Let me just summarize them for you really quickly. The first category we learned about was hormones, right? So hormones are signaling molecules. And they included, we discussed quite a few of them actually. They included things like insulin, which is responsible for food signaling and glucose use, ghrelin, which is another food-related thing, estrogen, which all animals have, not just females, testosterone, again, all animals have it, not just males, progesterone. So there are a ton of hormones. Oh, adrenaline. I showed you the structure of adrenaline in the lab, right? So adrenaline's another one. So hormones, they all have different kinds of structure, but the important thing is they're a relatively small molecule. and they tend to have a lot of rings that are sort of fused together. So I showed you pictures of different ones. So here's a benzene ring fused with another benzene ring. Oh, but I'm not drawing it very well. Fused with another benzene ring. Okay, well, you get the idea. My pictures are getting worse and worse because the computer is confused, right? So they're these big structures, right? Okay, so like just for an example selection, here's a bunch of hormones: estradiol, testosterone, progesterone. Okay, so they all do different functions. Cortisol is another one that's associated with like inflammation. You can see these are like carbon-containing compounds with lots of rings stuck to each other. Okay, so that's one common. These are all called steroids. These are ones that are steroids. Cholesterol is another hormone. It's not a steroid, but it is associated with a lot of these ones that are steroids. A quick look at the Wikipedia page about hormones shows you the steroid ones, kind of a couple of the ones that I just showed you. There's other hormones that are big, gigantic proteins, human growth hormone being one of them. Also, insulin falls into this category, I believe. So, and then you also have short chains of peptides. So again, this amino acids can come together to make hormones. And you can have even smaller ones, like norepinephrine is another small molecule hormone. So hormones can have all kinds of different shapes. But generally, for the purposes of this class, I'll just kind of limit any examples I might use to steroid hormones. Okay, so back to our discussion about the 4 different categories. A lot of slides open. Okay. So yeah, hormones is one. And again, all they do, they're signaling molecules. So they're things that tell you when you're hungry, when you're having a baby. All these kind of different things come from the human growth hormone that I mentioned, is what pregnancy tests test for, by the way. So hormones are not just involved in pregnancy and female things. Gentlemen need them as well, because that's how you know when you're thirsty or hungry or tired, or pretty much anything is happening in your body is signaled by hormones. Adrenaline appears when you're scared or in danger or, gonna fight somebody. Everybody has that adrenaline. So hormones are everywhere. And the next category that we talked about was lipids. Lipids are fats, and they fall into 2 categories. Either a fat, which is a solid material at room temperature, or an oil, which is a liquid. And we said that fats tend to be saturated. Now, we're not talking about in terms of saturated solutions. We're talking about in terms of having all the carbons that they can. So in other words, fats are alkanes only. Oils tend to be unsaturated, which means, oops, what just happened? Saturated. Oh, hey, that ended up in the right spot. So unsaturated means that it has double bonds or triple bonds. So alkenes and alkynes. Now, as we discussed, oils are better for you than fats, generally speaking, but both of them are necessary for life. So don't feel like you should never eat oil and fat. That is becoming increasingly clear that that is not a healthy diet, even though, beginning in the 80s, we were all told to eat non-fat everything. Turns out that's contributing to the obesity crisis. At least there's a lot of evidence for that. So anyways, fats, good oils, we need them. The next category that we learned about was carbohydrates, which of course most people have heard of. Carbohydrates, no matter what kind of carbohydrate they are, so, people say that like complex carbohydrates are better for you. Not really true. There's a lot of evidence for the fact that when you eat a complex carbohydrate, it just degrades down into glucose anyway, which is sugar. Sugars affect your hormonal system dramatically, whereas like lipids and proteins don't. So anyways, so the next category is proteins, which are really made out of something called amino acids, right? Amino acids are awesome because they are amphoteric. These mean the same thing. Okay. Basically, they can act as an acid and a base at the same time, which is cool. That reactivity is what makes them work. But proteins are the chemical catalysts of your body, and they also, of course, make up your heart, your muscle tissue, structural elements as well. So the final category is our information is stored in biological systems, and they're called nucleic acids. We talked about two kinds, RNA and DNA, and you should definitely know the differences among them. There's a nice slide in the lecture notes that summarizes that for you. Okay, so going back to this. Lipids have really long carbon chains. So this is probably a lipid because it's got all this carbon. And this would be an alkene because there's double bonds in it. Some of them are cis. Actually, they all are. So this is probably a really healthy fat, right? All cis is good. So this is some kind of oil. So the category here would be a lipid. It's probably an oil because we have double bonds in it. And then, to be specific, this one is called a triglyceride. So triglycerides are a really critical part of our biology. This is a polar head, we call it. So that's the beginning of the molecule. And the fat part is the nonpolar tail. Okay. So what's interesting is these can be used to make cell membranes. So if you have, I'm getting kind of sloppy here, but you know, if you have another tail and then your triglyceride, well, I did that wrong. That's what I get for freehanding things on a computer, right? So the triglyceride is made with the carbonyl right there and an O there, and then it's connected to the adjacent triglyceride, which also has carbonyl on it and an O. And then that's connected to the next one. So you get the idea. So if you had another long chain triglyceride like this, they can kind of put their tails together. And then the heads will point towards the center of the cell. So the cell would be out here. And then what we call the extracellular fluid is on the other side. So that's in between the cells. So this is what creates the membrane of cells, right? Long lipid tails with these polar head groups on the end. So triglycerides are one of the components that make your membrane. We call this a phospholipid bilayer. So lipid meaning fat, bilayer meaning two layers, right? So this is how it works. It keeps the water outside of the membrane, and that's good. That's good. It means we can control where water is going. And a lot of the sort of vitamins and things that we need can go through the fatty part of the membrane. Proteins, for example. There's a lot of nonpolar proteins that would hang out there. So that's the basic cell structure. It's about lipids in general, but specifically triglycerides are in there a lot, and a whole bunch of other stuff. So triglycerides aren't automatically bad. You need them to form cells. So keep that in mind when your doctor, is reading your blood panels. Now this one down here, this one's interesting. We have a 5-membered ring. So this makes me think about sugars. Sugars are rings with a whole bunch of OHs on it, right? But that's not all I have here. I've also got this group right here with all the nitrogen. As it turns out, when you have a sugar, right? So sugars look like either a 5-membered ring like this or a 6-membered ring, and then you got a bunch of another ring with a bunch of nitrogens on it. That is a nucleic acid. All right, so this chart is nice because it compares different kinds of nucleic acid properties. So we talked in class that the difference between DNA is that you're missing an OH right here. Why has it got that thing up there? Okay, you're missing an OH right there on that second carbon. So in RNA, it has an OH right there. That's one of the main differences between them. Also, RNA has uracil instead of thymine, so you can see that the difference between these is not really that big. It's just that RNA doesn't have the CH3 group right here, and DNA does, right? And in the middle we have adenine, guanine, and cytosine, and these 3 nucleic acids can be found in both DNA and RNA. So if we look at this structure here, I don't have any oxygen in it, and it has 2 rings. So this has an oxygen in it, and it's only 1 ring. That's got an oxygen, but it is 2 rings. This is 2 rings with a nitrogen coming out, so that's adenine. So this here is adenine. So that means this whole thing is a nucleic acid. That's the category here, so that's the answer to the question. But this is an adenine, and the sugar down here does have the OH group next to where the adenine comes off, so that would be a ribose. So that means this piece here is an RNA base where you've got an adenine attached to it. So that's going to pair up with the T, with the thymine. Alright, so the next one is cool because it's got a positive charge here and a negative charge over here, which means that this molecule could react with itself, which is kind of cool. Or another carboxylic acid group could come along and hook up with the nitrogen here and make another chain, another link in this chain. So this must be a protein. This must be in the category of amino acids. Each one of these little pieces, like here's a piece, that's an amino acid. Here's the next amino acid. And then here's our third one. So I know where to break them up because the carbonyl group is where the acid part comes out of. That's carboxylic acid, right? And the amine is the amino part. So this is each one of these residues. So this is a 3-chain amino acid. So what I look for is the nitrogen and the carbonyl group. And in the middle of the chain, the carbonyl group and the nitrogen are going to be together. So this makes what we called an amide. Okay. So amino acids have an amide linkage, which is also sometimes called a peptide bond because peptide is protein. Those two are synonyms. And so the peptide bond is this carbon-nitrogen connection that forms between different amino acids. Okay, and then finally our last one here. This is more rings that have a lot of OHs on them. So this is probably a carbohydrate. In particular, this particular one is a disaccharide because disaccharides have 2 sugars. So here's one sugar, here's a second sugar. And another thing to know is that sugars are aldehydes. Oops, I forgot the D. Let me just try that again. Okay. Sugars are aldehydes, and the trick is that when they're drawn in the ring, they don't look like an aldehyde, but when we pop this bond off, which happens, we call that a resonance structure. That's a phrase from 111. When that pops off, you've got a C double bonded to an O here with an H hanging off. So that's an aldehyde. Right? And so you guys tested aldehydes in lab with the tolerance test, and so this would have a positive tolerance test. Anyways, so that one's a carbohydrate. Now in this one we're going to take the same structures and we're going to determine what their intermolecular force is going to be. So looking here, we do have a polar group on our lipid. This polar head is there. But we have so much long carbon change that LDF is the most important feature of that molecule. Okay. For B, that's our nucleic acid that we're looking at. We got a lot of OH, we got an NH, and then these nitrogen ones are good sites to receive hydrogen bonds. So the dominant force there is going to be hydrogen bonding. Now amino acids are funny, so we have to look at each individual amino acid to decide whether it's polar or nonpolar, because the backbone is relatively polar, but whatever comes off can often be really big. So these are those R groups that we were talking about, right? That makes it whatever amino acid that it's going to be, and those R groups are what give proteins their functionality. So like if I have something that's going to be in a cell membrane, in a fat, it's going to be pretty nonpolar. If I have something that's going to exist in the cell where there's a lot of water, it's probably going to be relatively polar. Okay? Some amino acid residues, some peptides, are both. So the inside part might be really nonpolar and the outside might be really polar. Okay? That happens quite a lot. So I have to kind of look at this and decide what it is. So this is going to be a polar residue because you have a dipole. This amide group down here is a dipole and a hydrogen bonding thing. So that's going to be a really polar molecule. It's going to like to hang out with water a lot. Over here you have a charge. That's definitely going to... Sorry, I yawned. That's definitely going to be very polar because it has a positive charge, and water is really attracted to charges. So far, this is a pretty polar amino acid residue. And finally we have this one. Now I don't remember off the top of my head how carbon and sulfur are related, so I'm just going to look at an electronegativity table. Okay, so here we go. So sulfur is listed, oh I can't read it. Here we go, here's one we can read. So it's 2.5, oh, and 2.5. So at least according to one of these electronegativity tables, carbon and sulfur don't have a difference in electronegativity. I just want to check that with a couple more. 2.5, 2.5, okay. So it seems to be true, at least with a quick glance at the internet. I'm skeptical, because I'm always skeptical of the internet, but it seems to be that the carbon-sulfur bond is not very electronegative. Now, I think this sulfur would probably be a bent configuration, which might create a small dipole, but mostly this residue is probably nonpolar. So we have things that are polar, polar, and nonpolar. I'm going to go with majority rules here. So we have two things, basically. Two different hydrogen bonding sites here, one here, and a dipole, as opposed to like one chain that is just LDF. So I'm going to say that hydrogen bonding is the most dominant thing because we have so many of them, 3 different hydrogen bonding sites. And also it's stronger than the other forces. And finally D, our disaccharide has so much OH all over the place, so that is also hydrogen bonding. So we're going to notice that 3 out of the 4 categories tend to have a lot of hydrogen bonds in it. Only fats don't, which means fats don't mix with water. So that's interesting. But you knew that because, you know, vinegar and olive oil don't mix together. Oh, so lactose is a really interesting sugar. It comes from milk, right? Lactose is in milk. And it's interesting. People don't think of sugar as being in milk, but it is. It absolutely is. Unless, of course, you're drinking lactose-free milk. But even then, they've replaced it with other sugar. Okay? In fact, non-fat milk has more sugar in it than whole milk. All right? So there's this balance in the products we buy. When the non-fat dieting craze started in the 80s, 70s and 80s, companies had to compensate for missing the fat in food by replacing it with something, and protein's expensive, so they mostly replaced fat with sugar. And it's believed that that replacement in so many of our products has caused a lot of the obesity problems. So the next time you're at the grocery store, I dare you to compare non-fat milk and whole milk and see which one has more carbohydrate in it. A spoiler alert, it's going to be the non-fat milk. It always is. The same thing for your yogurt, like all your dairy products. So that's a fascinating bit of nutritional history. At any rate, here's lactose. This is the sugar that's in milk. So what we're going to do first is we're going to start out here at this oxygen and then make our 6-membered ring. Okay, so oxygen first, and then there's 5 carbons attached to it, and that's how we get the 6-membered ring. Okay, and then next to the oxygen on this ring we have a CH2OH that goes up. The direction is really important because if it was flipped downward, that would actually be a different sugar. And then we got 2 OHs that go up, and then I think the third one here goes down. So that's the first sugar in lactose. And then we've got this oxygen connection between the disaccharide is connected with an oxygen. So we're going to draw that. And we have another 6-membered ring where the oxygen in the ring is 2 carbons away. So we're going to go 1, 2, oxygen, and then finish up our ring. And then CH2OH here going up. Our OH go up there, and then 1 down in the middle. So this is lactose, okay? And again, the OH direction is important. And then it says draw where the lactase enzyme is going to break. So in order to, it says to break lactose, which is a disaccharide, apart. So this disaccharide is the key, right? When it says disaccharide, I have 2 sugars. Saccharide means sugar. So my lactose, well, also OSE means sugar as well. So anytime you see that, be on the lookout. It's going to have carbs. So OSE means sugars. I have 2 of them, so I have to break them apart before I can metabolize anything. So our body can only break down single rings at a time, so we got to break this. So your enzyme lactase is responsible for doing that. It holds the molecule in exactly the right way. It must have exactly the right shape to hold it, and then we facilitate breaking this bond, so we have 2 monosaccharides, which are easy to digest. If you don't have lactase, then you can't break this molecule apart, and it will cause your tummy to get upset, along with like a whole bunch of other things, depending on the severity of your allergy. People with less lactase have a more severe allergy. Also, as we get older, people tend to have less lactase present in their gut anyway, so you can become lactose intolerant as you age. Okay, so this last question on this page: what biomolecule is the enzyme most likely made from? Well, it's an enzyme, so it's probably a protein, which means it's made from amino acids. The only other thing enzymes can be made out of is RNA. The most common example of that is an enzyme called the ribosome, which is where our protein is made in a cell, and it's ginormous. It's very, very big. You can sometimes see it as little specks under a microscope. It's a single molecule that's really big, which is cool. All right, and then this one is about the, let's see, what IMF would we expect lactose to have? So we look at this picture. It's a carbohydrate, which means lots of OHs, so it's going to have a whole lot of hydrogen bonds, which tells us that the enzyme active site where the lactose would be held must have some complementary hydrogen bond acceptor. Okay, so what, oh, I just said the word active site, and look, there's a question about it. The active site is where the chemical reaction occurs in a protein. Okay, so it has to have the exact correct shape, and it has to have kind of like complementary IMF, right? So they have to match each other pretty well. And so we had images of enzymes and how they work in our lecture notes. We called it the lock and key mechanism. So take a look at that. Okay, so this is this last question on our biochem homework, which is also the last homework you have to do in chemistry, I hope. If we pass, that's good. It's been super fun for me. I hope you've learned something. I'm sure you have. As you are preparing for your final exam, be sure that you understand this material and you can identify how different IMF relate to different biomolecules and how their physical properties might be different as a result of that. That's kind of the point of this chapter, really. Okay, so we know the 4 categories: amino acids, carbohydrates, lipids, and nucleic acids, and we can identify them based on a picture, and we can tell a little bit about their forces. And yeah, so I will see you in class. If you have questions, you know you can always reach out.